I'm going to talk about the 100,000 Genome Project, in particular the role that Genomics England plays in it. I should say right now, because it's important for my next slide, that Genomics England is a company. For those of you watching this in California, that means a corporation. That is an organization who is governed by their board, in our case, a very distinguished board with a majority of independents, one of whom is John Bell, who you've just heard. Therefore, we're not like the NIH or the MRC, which I used to chair, integrated part of the public sector, but we're a company, but it happens to be owned by the government. And so normally, we are completely separate from all that sort of stuff, but somebody got to hear that I was going to be talking here this afternoon, and indeed on another event in a week or so's time, and sent me a longish letter running to some 15 pages, describing the rules of PERDA. Now, I don't suppose all of you, particularly in California, understand what PERDA means. It is a term that was originally, it's an Indian term, for the state of incommunicado that the wives of Maharajas go into once they join the gang in the place they go to. And not allowed to talk to anyone at that stage. For a century and a half or so, the English civil service has adopted this. So the moment Parliament stands down, is prorogued by the Queen, and there are no MPs. There are only candidates to be MPs. The public sector is not permitted to say anything in public which could be taken as favouring one or other of the many candidates. And I say all that because, if I point to, there are two gentlemen on this. One of them is a prospective parliamentary candidate for Witney, and the other is prospective parliamentary candidate for Surrey Southwest. And doubtless, I fail in my mission by not putting up pictures of the other esteemed candidates who are up for election in those two constituencies. But I will relate the precise fact, which I'm allowed to talk about, that these two gentlemen were big supporters in getting our project going, and a very important step forward in the topic of this conference that I think they set in motion. So what exactly have we set in motion? We set in motion a project which I think probably slightly different to Eric's project, is not designed as a research project. It is designed specifically as a project to transform the National Health Service. And so it is not designated as a research project. It has these three principal goals. One, if I can press that. Oh, there we are. That one. Increased insight, i.e. finding new treatments and devices and diagnostics. As I've just said, advancing genomic medicine, precision medicine in the National Health Service, and importantly also a thriving private sector alongside what we're doing. But crucial to all of those, we recognize, is this question of positive public support, because in order for us to do our job, we need our patients, we need the citizens to be absolutely behind giving us their data so that we can give back to them the therapies and diagnostics which can come from it. Public sympathy is a fickle thing, and there's plenty of evidence about this. If the public get on the wrong side of any scientific issue, it takes a generation to reverse that. And it is crucial at this stage in the development of genomic medicine and precision medicine, where we are collecting patients' data without immediately being able to give them a therapy which will help them, that over this period we pay every attention to keeping public sympathy on side, which means to a degree providing, as I shall show as I shall show in a moment, some constraints on ourselves, which are not immediately helpful in pushing forward the project, but do help carry public sympathy. So, I won't be able to tell you, because of PERDA, the whole story of the early days of this project, which was quite fraught in many ways. Because following the announcement, there was a period of 6 months or so where there was a lot of argumentation about what direction the project should take. It was settled that it was a project based upon whole genome sequencing. And that's what we are, and I won't explain to this audience why that decision was taken, but to say it is taken, and that's what we're doing. So, what therefore are the distinguishing features of the project? Well, first, it is structurally integrated into the National Health Service. So it is not a pharmaceutical trial or an experiment or experimental project of any sort. It is structurally integrated into the National Health Service. It is based not upon the worried well, but upon a selected cohort of patients chosen because of what their conditions might be able to reveal. Those patients are very positively consented, so we tell them up front exactly what we intend to do in its full dimension. So we hide nothing and make a point of saying the things to them that they might object to, in order to make sure that we are very sure that they have positively consented. I'd have to say that with patient groups, there's never a problem with that. The problems we have are with people who have a broader issue to do with privacy in health. We, of course, are setting up a state-of-the-art sequencing center, more about that in a moment, and crucially, we're setting up a safe haven. That is a critical part of the deal we have with patients. And there you have it, the three things that patients we find particularly relate to, which makes it possible to carry patient support, to carry public support, is the fact that it is integrated into the health service, that all the patients have very positively consented, and we are doing this thing of putting all their data into a safe haven. That means that we do not provide the data to outside our safe haven. What we provide is a reading library and not a lending library. That, of course, has its logistic consequences, which are not ideal in a research world, but it is an important step that we need to go through to carry public confidence as we get over the hurdle of it becoming accepted that there's real benefit for the public coming from this program. As soon as there's real benefit for the public coming from this program, I'm absolutely confident that the public will fully support it. But we have to go through that period. Right. Now, the whole life cycle from patient through a genomic medicine process back to the patient getting something from it is a process involving many, many, many steps through very, very many different organizations. And what I have learned in the time that I've been engaged on this project, and reminding you of what Peter said about me, I'm an engineer by background, and I've done most of my career in the era when the data revolution was running through numerous industries. What is apparent to me, looking in on the industry which we are now engaged with, is how much of a cottage industry it relates to. That every step on this process involves normally principal investigators in control of their own lab, doing their bit of DNA extraction or their bit of pathology, and so on. And every step of it is critical, because if you don't do it in a completely consistent way, you introduce artifacts, and that's all right, because actually the state of sequencing technology we're in at the moment, artifacts at some level are inevitable. But if you're going to be able to get a coherent result out of it, it's important that they should be the same artifacts, not lots of other different artifacts. And so it is crucial in getting the process working at the level of technology we are right now, to industrialize the process so it is done in a precisely consistent way right through the whole of the cycle, going through the various organizations which are involved in it. So, to do this at the scale of 100,000 whole genomes is a colossal undertaking. Having seen Eric's figures earlier on, to give you an idea, the whole cost of this is north of £300 million, $450 million. And that is even after you've done the industrialization to get the costs into a realistic regime. But it inevitably involves not just one organization, Genomics England, and in fact all Genomics England do essentially is to coordinate this whole process. It involves critical partnerships with other people who are well placed to orchestrate and undertake the parts that they're engaged in. And let me run through those. First of all, the clinical partnership. Genomics England, who after the disaggregation of the old NHS and the taking of responsibility in the new nations, Scotland, Northern Ireland, and Wales, England falls to NHS England, and England is responsible for some 55 million patients within England. Their role is to undertake the processes at the front and the back end of our cycle. The processes of finding the patients, consenting them, extracting the blood, extracting the tumor samples, extracting from that the DNA, doing the logistics of barcoding them and all the rest, transporting them that to our biorepository, and at the back end receiving from us the output from our sequencing, doing the necessary verification is required, and handling the patient. That's what and to do that, Genomics England have run a process which is not the sort of thing which we could have done, and indeed did do during our pilot program, which was to gain volunteers and enthusiastic hospitals to join with us. It was a commissioning process, i.e. part of the integrated way, the normal way in which work is done within the NHS, which is a commissioning process. It was a competitive process. Hospitals all over the hospital trusts all over the country were invited to bid for a number of mandates. It turned out to be 11 mandates to become genomic medicine centers. This turned out to be enormously important amongst the hospital community. Why? Because hospitals, of course, the trusts are independent bodies. They have their own finances to manage, and for that they require to position themselves to get business, commission business in through the door. The it is clear that the direction of travel in the NHS is through specialisation. The direction of travel is that there will be fewer specialist organisations across the country specialising in different areas of medical science. The identification of genomic medicine centres was regarded by the community out there as a first step in that direction. So it became crucial to major hospital trusts, particularly those that were part of the academic health sciences networks, to make sure that they were part of that regime. And they all tried extremely hard to make sure they got into it. This is very good news from the project because it means that we have as partners the 11 genomic medicine centres who are fully committed to do that part of the of our 100,000 genome project, which if you look at similar ventures which have been tried around the world, have found in many ways most difficult, which is acquiring patients and getting quality data from them. And that, of course, is quality data, not just in terms of the samples, but also of the clinical data. And I echo John's points. The clinical data arena, it's all right saying that hospitals have invested in electronic medical data systems, but that only, says like, most of the country have got motor cars. Well, that's true, but they don't all do the same things. And for us, given that importance of coherence that I described early on, it's crucial that we industrialize that data collection so that we get exactly the same every time. And one of the key things which we in Genomics England have been involved in is providing data models to the genomic medicine centres, which describe precisely what we need and defines precisely what we need, so everybody knows when they're giving us the data, they know exactly what we want, and we can check that we get exactly what we think we've got. Right, going to the next major partnership, and that is the sequencing. We ran a competition across the whole world. We provided samples to anybody who said or thought they could do whole genome sequencing at a reasonable volume. We had 6 offerers for that. Only 2 eventually succeeded in passing the test, and we chose Illumina. We chose Illumina because Illumina had done more of this than anyone else and provided at least as good an answer to the test. But actually, frankly, more importantly, Illumina were absolutely committed to work with us to progressively improve what was possible in whole genome sequencing. So we got to the point whereby after we'd done 100,000, we had actually proven that whole genome sequencing was going to be viable and useful in routine medical care. That's the partnership that we set up. We are almost, nearly, in this new sequencing center that is being built courtesy of the Wellcome Trust on the Sanger estate. And we are working extremely closely with David Bentley and his team in Great Chesterford, not just on the sequencing side of this equation, but actually on the front end of that, working with us to make sure that we improve the sample inflow and eliminate so far as possible the artifacts that we're seeing in it, and also working with us on the output side so that we improve the process, the software processes for the analysis and interpretation of the data, and more about that in a moment. Right. The other thing we did, one of the other things we did when we set up this project was to run an international competition as to who could interpret whole genomes. Provided a suite of whole genomes out there, and we expected, rather like the sequencing bake-off, to get a series of results which would center around a few people who clearly do it well, and there'll be a spread of others. What we discovered was in the interpretation arena, there isn't any gold standard. There is a huge variety out there. It's the Wild West still in terms of where the science is. So we changed tack from that and said, well, what we have to do in the interpretation space is call in all the best minds we can find all over the world, and particularly, obviously, in the UK, to work with us. And so we put out a call for that. It's been answered by a large number of teams were formed. We wanted to have essentially one team per disease syndrome or per a generic attribute that we wanted to look at. I say we're expecting around 2,000 scientists. Actually, it's more like 4,000 scientists at the moment in terms of the scientists who put their names forward to participate in it. And we are setting up a number of domains, say one team, one international team per domain to work with us. And here we have a bunch of some of the domains that are already being set up. This is absolutely central to what we're doing in the 100,000 Genome Project, because it is the mobilization of that great resource out there in the clinical and scientific community behind the data set that we're collecting, which will reveal the insights which we can build into the automated tools from which we eventually want to emerge from this project so that we have the basis of a diagnostic which is usable in routine medical care. Right, so what are we in Genomics England doing? Well, the key thing we're doing in Genomics England is providing the central resource on which all this will stand. So we're the bit in the dotted blue lines in the middle here. We're providing the informatics infrastructure and the bio repository. In terms of the informatics infrastructure, as the 2 points worth drawing up. One is we have a firm division in the infrastructure between a management system, which is high security. That's the area that holds the only area that holds identifiable data, and that is accessible essentially by Genomics England and by the clinicians that have provided the data and no one else. The data is anonymized there and then passed down to another data system, which is our research system. And in here, we provide embassies for all our various users to participate. We provide a software infrastructure, which we think of as being app-based, in that because we know at this time that there are no gold standards out there, we will provide a variety of software tools and techniques available to users. And over the period of this project, we hope and believe that what will emerge are clear winners, at least winners per disease syndrome that we're working on. And the last of our partners is the commercial world, which we have brought on board. We separate it between the pharma community, the biotech community, the smaller biotech community, from whom many of the insights will be discovered, because pharma is not, on the whole, well focused upon genomics at this moment, and much of the innovation we think will come from the biotech community, and finally the bioinformatics community, who we tend to think of as platform partners. So in terms of the platforms that we've got, we think of it in terms of 4 layers. There is a layer 1, which is our reversionary layer, which we use for getting as quick a product we can in order to provide diagnosis back to the patient. We have layer 2, where we have multiple interpretation algorithms available for trialing different approaches. We have a layer 3, which is where the SMEs have been invited to participate, and we'll have probably quite different answers to. And then we'll have the rich heterogeneous inventory of tools and techniques which will come from the GSIP community. The pharma community we see as being, we see in the first instance as forming a single pre-competitive consortium, and we have 10 or so companies already signed up to this, who will work together during 2015 to help amongst themselves find how best to go about using this new resource which is becoming available. So those are our principal partners. There are many other partners who we're working with, and I don't have time to run through them. We're working with the regulators because as you introduce genomic medicine and precision medicine, that is a whole new model, and we don't want, and it will be not as efficient as it could be if we stick to the previous regulatory regimes, which, as we know, take many years to pass through. And a whole bunch more, which again I don't have time to go through. There's the multi-omics approach to cancer, which we're working with partners here in Oxford on, with molecular pathology in order to improve our ability to get good samples, particularly from solid tumors. Complementary diagnostics in order to get us into epigenomics, transcriptomics, and proteomics, etc. Longitudinal data sets. We're working with the Health and the HSCIC, who hold the national health data and social care data, in order to get longitudinal data so we can follow our patients over many years, and technologies like circulating tumor cells in order to be able to follow the progress of cancers. These are all separate projects which are subject to partnerships, which we are working together with those partners to deliver. So, what we can expect for patients, we can expect patients to get feedback on pertinent findings, and also on, if they opt for it, on specifically pathogenic findings. For research collaborators, there is the access to this phenomenal data set, the ability to work with our pharma partners, and the opportunity to engage in translation. For our commercial partners, it's the inverse of that, clearly working with this huge community of researchers to help them develop products and diagnostics, and of course the general public shall eventually see the benefit of it. So that's how we intend to deliver those ambitious goals we set ourselves as we started. I end by just complimenting the organizers here on having set up this timely event. I, as you probably understand, I started my life and accidentally came in to the field of computing and software at the end of the 60s. It was an extremely lucky choice because I rode through the latter half of the 20th century, that tidal wave of opportunity which came out of the microelectronics revolution, and it changed the world, as we all know. What we're about in this room is the equivalent for the 21st century. It is what we are able now to discover by applying genomics and precision medicine to the health arena, which will transform the living experience of the human race in the 21st century. it is the same, if not greater, change in civilization, which we'll see. And I congratulate everyone from choosing this field to work in. Thank you.